Hi, we are going to discuss very important questions in today's lesson. This question is based on the structure of the proteins. So this is again Unit 1, Chemistry of Life, Biological Molecules. Under that, we are going to learn about protein molecules, right? Okay. First of all, we're going to learn a structure of protein. So we call it protein molecule because molecule is something that is built up of a combination of different elements, and they are bound chemically to make a molecule. So protein is a molecule, and protein is a slightly different molecule. It's a very big molecule. So we call it macromolecules because macro indicate a size of the molecule, large size of molecules. Also, we can call it a polymer. Polymer is a large molecule which is built up of combination of many repeating units. The repeating units are called monomers. So it is just like we can imagine a house wall is built with bricks. So house wall is a protein. The bricks are monomers. Now the monomer of the protein is called amino acids. So there are 20 different amino acids we have learned. All the amino acids they have a common structure like this. Carbon. So carbon can make 4 bonds, 1, 2, 3, 4, because it has 4 electrons in the last shell. So these are the valence electrons. So these electrons can make a bond with carboxylic group COOH. And this one can make a bond with the hydrogen, and the third one can make bond with amino or amine group. So this has a nitrogen and 2 hydrogen. This black text indicate the common structure of the all 20 amino acids. Now the blue one, I'm going to write here R. R is a variable group. So R is changing from one amino acid to another amino acid. So this is the basic structure of an amino acid. And again, the important thing we should highlight, this one is common to all amino acid. And R group is changing to a different amino acid. As a result, we can find many different types of R group. For example, a polar, maybe a non-polar, hydrophobic, hydrophilic. So there are different types of R groups, and they have different properties. So these properties are very important to decide the chemical structure and function of the proteins. Okay, right. Next, we are going to learn how amino acid join to make protein molecules. So I'm going to show you here 2 amino acid joined together to make the protein molecule. Okay, here I'm going to show you again one amino acid, carbon. And this side carboxylic group, I can expand the carboxylic group like this, and this side is the amine group, amino group, hydrogen, and this is the R. Now here comes the second amino acid, carbon, amine group, carboxylic group, H, and I going to show you a R group differently, R2. So then first one may be R1. Okay. Now when we have a two amino acids, so this OH and one hydrogen of this can combine. So when they combine together, resulting molecule is water molecule, H2O. What is left now? This OH is joined to make a water molecule. One hydrogen is joined to make water molecule. Now in this case, C double O is here, and from the other side, nitrogen and one hydrogen left. So then bond is making between carbon and nitrogen. So this is the bond. And rest of the thing remain as same, no change. For example, C, H, R1, amine group. And the other side, same, carbon, hydrogen, C double O H, and R2. Okay, this bond is very important now, and this bond is called peptide bond. Peptide bond. The reaction is called condensation reaction. Condensation reaction. Because one water molecule is formed, as a result, the name is given condensation reaction. And we can also break this bond. So I will also show this here, like this. If we add a water molecule, then we can break the bond. So if they break the bond, we call it hydrolysis. Peptide bond can be hydrolysis by adding water. Peptide bond form when one water molecule is formed in condensation reaction. Now, this is the dipeptide, right? So dipeptide means two amino acids joined together to make the peptide bond. Okay, now just to summarize about the peptide bond, when two amino acids join together, a peptide bond is formed, and one water molecule is also formed. This is called condensation reaction. So we can also highlight the peptide bond. What is the chemical structure of the peptide bond? Carbon, oxygen, and then this is the bond, nitrogen and hydrogen. So this is the peptide bond. This side we have a one amino acid. This side we also have one amino acid. And we can also break this peptide bond, which is called hydrolysis. Just adding a molecule of water, we can break this bond. Right now, you see here two amino acids. We know that to make a protein molecule, there are hundreds, thousands of amino acids joined together. So when they join together, I can show you some amino acid. So let's say this is one amino acid, 2, 3, 4, 5, 6, 7, 8, 9, 10. So between these 10 amino acid, we can also find the peptide bond here, 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 here. So once it formed, we can also notice one important thing. This end, uh, the amine group is here. This end, COOH is here. Carboxylic group is here. Because this carboxylic group is not involved to make the bond. If one amino acid join from this side, then again it is going to involve to make the bond. So this side is a C terminal, this side is an N terminal. So we can always find in a protein molecule these two N. After that, we have learned this peptide chain. We call it polypeptide chain. Poly mean many, peptide chain. They're going to fold. The folding is resulted between these R groups. So we know each amino acid has R group. And these R groups are different. So some can react with each other and some cannot react with each. Some cannot react and some have hydrophilic, hydrophobic and polar and non-polar property of this R group. As a result, we can find secondary structure, tertiary structure and quaternary structure. So polypeptide chain is considered as the primary structure because primary structure is the order of the amino acid or sequence of the amino acid. Order of the amino acid or sequence of the amino acid. Secondary structure, we have learned there are two forms. One is alpha helix, other one is the beta pleated sheets. The name is given based on the protein folding and we can find only hydrogen bond. Tertiary structure is the 3D structure of the proteins and we can find complete structure of the protein, final version, and this has other type of bond, hydrogen bond, ionic, sulfide, and hydrophobic, van der Waals bonds. Tertiary structure is slightly different, so we can consider tertiary structure as a combination of few different quaternary structure is a combination of several tertiary structures. Here you can see the quaternary structure has one tertiary structure, two tertiary structure, 3 and 4, and they are bind with the chemically. Okay, this is the quick revision of the proteins. Now this knowledge you need to use to answer